Hello students, welcome to the class of Botany. Today we will be dealing with your transcription. In the last videos and the last lectures we have already finished our application. If you really want to know about it, so what you can do, you can just subscribe to the channel, and then what you can do when you have subscribed, so generally what happens whenever there is an update with respect to any particular topic, then you will get the notifications, okay? And yes, if you want to search about it, you can just type in the topics which are a bit not easy for you to understand. So with this note, let's start with your transcription process. I have just broadly written here what does is your transcription. Transcription, this detail we have already done in the replication when I was teaching you about a central dogma. But still the people who have just joined in or just want to see this, transcription is a process. Transcription, I'll just write it down here. I'm writing down the definition of transcription. Transcription is a process in which your double-stranded DNA is converted into your single-stranded mRNA, because this mRNA, it is supposed to make proteins. This mRNA, it is supposed to make proteins, and this transcription process generally needs your DNA dependent RNA polymerase enzyme. DNA dependent RNA polymerase enzyme. Later in the lectures I will be telling you which polymerase enzyme do we need for the synthesis of mRNA. And this whole structure or this whole function, it takes place inside nucleus. It takes place inside nucleus. Once DNA gets converted to mRNA in the nucleus, the mRNA will move out of the nucleus in the cytoplasm in order to synthesize proteins, or you can say amino acids, by the process known as translation. Okay? So this is the core basic of your transcription process. So now let's start with the process. Students, when I say about the process, transcription process was initially, along with some fellow co-workers, it was given by Kornberg. And you should not forget, this was the same person who is known to give Kornberg enzyme. It is the same, he's the same person who gave the Kornberg enzyme. When I'm saying Kornberg enzyme, I'm talking about DNA polymerase 1. DNA polymerase 1. Transcription, the process in which your double-stranded DNA is converted into your single-stranded mRNA with the help of an enzyme, DNA-dependent RNA polymerase. And yes, it is given by Kornberg. Kornberg was the same person who gave Kornberg enzyme. That means I'm talking about DNA polymerase 1. Remember, the DNA polymerase 1 has that proofreading material in which it removes RNA primer. If you want to know about it, please go back and look for the replication of DNA topic. Alright, so now let's move ahead with your transcription process. This is what we all know, that yes, transcription is a process in which generally there are 3 steps. First one is initiation, elongation and termination. That I will deal with right now. But before that, when I'm talking about a transcription, there is a unit, there is a segment of DNA which is known as a transcription unit. Let's see what does that transcription unit consists of. So transcription unit will generally have a promoter region, set of structural genes which are supposed to be transcribed, and then finally a terminator. When we talk about your structural genes, these are the genes which will help in the synthesis of mRNA, because they will be containing coding and non-coding regions. Coding and non-coding regions. Okay. So now when I'm talking about your promoter region, I won't you guys to pay attention to this. These are the terminologies which are very important. When I'm talking about your prokaryotes, prokaryotes will have a promoter region. Prokaryote will have a promoter region, but there are certain conserved sequences present in your promoter region in prokaryotes. Simultaneously, conserved sequences are also present in eukaryotes. They also have conserved sequence. When I say prokaryotes, they have 10 base pair upstream conserved sequence from the start point. From the start point, 10 base pair upstream. Simultaneously, in your eukaryotes, 20 base pair upstream, they will have a conserved sequence. They will have a conserved sequence. So, in case of prokaryote, the conserved sequence are known as TAAT box, or you can say Pribnow box. Pribnow box. When we talk about eukaryotes, eukaryotes will have a very famous Tata box. The reason why they are known as Tata box, because they have a above 20 base pair upstream, they will have a 7 base pair long conserved sequence. 7 base pair long conserved sequence. That sequence will be TATATAT. So the name has been given Tata box, which is also known as Hogness box. In case of your prokaryotes, this will become a Pribnow box. Okay, so now let's start with your topic of transcription. So now when we are talking about your transcription process, as I initially told you, that transcription process will have certain steps. So let's talk about the steps in detail. First is your initiation. Students, you need to pay attention to this because I will be telling you in the form of diagrams here. Say for example, this is a sequence of DNA which is supposed to be transcribed, in which the transcription is supposed to happen. So let's identify the regions here. So this will be your promoter region, this will be your terminator region, and this will be your structural genes, which are supposed to be transcribed in order to synthesize mRNA. Of course, your transcription initiates from 5' direction to the 3' direction. Okay, in your NCERT books, you might see it like this. Okay, so in your NCERT it would be like this. So in order to simplify, I have just made a simple outline in order to make you understand that how the transcription is gonna occur here. So now let's see what happens here. So during the initiation process, of course initiation means the transcription is supposed to start. But in order to start transcription, what I need is that I will be needing RNA polymerase. I will be needing RNA polymerase enzyme. So generally, RNA polymerase are of so in order to understand this, the first thing, RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Let's see. RNA polymerase 1 is the one which will make your rRNA. Polymerase 2 is the one which will make your mRNA. And polymerase 3 is the one which will make your tRNA. Majority rRNA, ribosomal RNA, messenger RNA and transfer RNA. Along with mRNA, they will also synthesize hnRNA, which is your heterogeneous or heteronuclear RNA. Then you have your tRNA. Along with tRNA, they can also synthesize small nuclear RNAs. But what is the most important thing that we need right now is this. So which RNA polymerase I'm going to use here? I'm going to use RNA polymerase 2. I'm going to use RNA polymerase 2. So let's see what happens here. Okay, so generally what happens, I'm going to use different pens in order to make you understand properly. Promoter is the region which provides the attachment site for, let me make RNA polymerase like this. So this is the region, promoter site, which makes a site for the attachment of RNA polymerase 2. This is your RNA polymerase. So the RNA polymerase attaches to the promoter region in order to start the process. But the transcription process, agreed, it needs RNA polymerase 2 in order to start transcription. But RNA polymerase 2 itself cannot start the transcription process. So in order to do so, it needs some help. And the help would be sigma factor. And this sigma factor is also known as initiation factor. This sigma factor is also known as initiation factor. If you can see, this has attached here. So the moment, this is the RNA polymerase 2. So the moment it attaches to RNA polymerase, it will push RNA polymerase. And then once it is pushed, the RNA polymerase will start transcribing the structural genes. Let me show you in another diagram what happens. This is what we are talking about. Structural genes are there. Now please pay attention. The moment it starts the transcription process, it forms a bubble. It forms a bubble, and this bubble is known as transcription bubble. And this is known as transcription bubble. If you can see, I'll just draw it with the black so that you guys don't get confused. It forms a transcription bubble. You see this? This is still attached here. But what I get is that once it starts the transcription process, sigma falls off. Sigma falls. Why? Because sigma was supposed to only push it, not to be there during the whole process. So the moment it pushes, what happens? The sigma factor, which is the initiation factor, it falls up. And normally the RNA polymerase 2 will form the mRNA. It will start during the transcription. Now let's see what happens in the next set. Sigma is not there. You know that the transcription bubble is made. Transcription bubble will tell you that yes, What has started? Transcription process has started. So let me rub this off for you. You see this RNA polymerase is still here. RNA polymerase is still here, but what will start? It will start making your mRNA. It will start making your mRNA. Okay. Now this transcription bubble is there. Let me see, I told you that during the structural gene transcription, you will have some coding strands or non-coding strand. Let's see here. So this strand is taken as your template strand. Template means, of course, the parental strand which will help in the synthesis of a new strand. So this is the template strand, which is also known as your non-coding strand. Why a non-coding one? If this is the template strand, then it should be 100% coding. I'll tell you the reason behind it. And this one is your non-template strand, which is also known as coding strand or a sense strand. Sense strand, coding strand, non-template strand. Now what happens? When this RNA is being made through a transcription process, say for example, these are the genes, they will get the code A, T, T, A, G, say for example. Now, using this template, this double-stranded DNA, the other one will of course make a copy, T, A, T, G. Of course, this is a template. So using this, a normal DNA will copy the messages like this. But mRNA, which is being synthesized here, it will use this strand in order to copy the nucleotides. Copy means they will exact take all of the information, but instead of T, what they will add? U. So in order to copy the information, they will not do all the hardship work. Just what they do, they will generally copy it from this strand, and only everything remains the same because they are not doing anything else. They are just copying it. But it is mRNA. That is the reason. Instead of T, only U is there. Rest remains the same. So this transcription bubble continues, and eventually at the end, this transcription bubble will at the end reaches termination. So when the terminator region is there, so once you see a bubble here, that means it is about to end the transcription process. So it cannot happen like that. Remember, in order to initiate the reaction, they needed initiation factor, which is your sigma. Simultaneously, they need a termination factor, and that termination factor is known as rho. So a rho is going to come here, and once this bubble comes in contact with the rho factor, which is your termination factor, then what happens? mRNA is here, RNA polymerase falls off, and the transcription process stops. So this is the whole process of your transcription, which has taken place inside the nucleus. But you need to know this mRNA which has been made, it is not ready to go outside the nucleus and ready for translation process. The reason is that because this mRNA which has been synthesized, it is full of functional and non-functional regions which we need to get rid of. That is the reason this functional and non-functional regions are not present in your prokaryotes, But yes, archaebacteria is exception, and this functional and non-functional regions are present in your eukaryotes. Functional regions are known as exons, where the non-functional ones are known as introns. So we need to get rid of them. So about this, let me write down a quick review of it, that what we have done. I would say a recap. So when I say a recap, let's do it like this. This is a transcription. So let's see, what do we need in order to do the transcription? What I needed? I needed RNA polymerase 2. What I needed? I needed sigma factor, which is my initiation factor. Then I needed structural genes. And then what I needed? Termination factor, which is your rho. And yes, of course, how can we forget about this? Promoter region, terminator region. So from the transcription, that means your double-stranded DNA molecule has been converted to your single-stranded mRNA molecule. So this mRNA molecule, which has just been synthesized, it is not ready to go out of the nucleus to do the translation process. In order to do so, it needs to get rid of introns. And it is done with the help of a process known as splicing. So this splicing topic will be covered in your next upcoming lecture, and hopefully this lecture has been helpful for you. And you have any doubts regarding the transcription process, what you can do, and the same field where you have subscribed the channel, you can go down to the comment section and you can drop your questions there. Thank you so much, students, for watching this video. Thank you.